Hi all, welcome to this virtual presentation. Foremost, I wish everyone safety and request you to take necessary precautions to stay safe in these times of pandemic. Today, we will be making a presentation around biophysical screening and fragment-based drug discovery at Jubilant Biosys. The outline of our presentation comprises of brief information about Jubilant, followed by specific details around what we do in the structural biology department. We have information lined up around biophysical platforms, the use of SPR in further detail, and of course, we talk about how we go about fragment selection and fragment screening using SPR, while ending the presentation with a summary of this discussion. All in all, we expect to take anywhere between 25 to 30 minutes to complete this presentation. Starting with Jubilant Life Sciences, which is our parent company. This is a company which has been in existence for well over 40 years and has an annual sales turnover of almost $1.2 billion. As on date, we employ more than 14,000 people, and of those, 1,300 people are based out of North America. The Jubilant Life Sciences business is split into three key business verticals: Jubilant Pharma, where we have generic business and specialty pharmaceuticals. Within the specialty pharmaceuticals business, we operate and own Hollister Steer, which is a CMO doing sterile fill finish for pharmaceutical companies. And we also have a radiopharmaceuticals business. Jubilant Life Sciences has been the bread and butter business, and this business operates into three segments: specialty intermediates, nutritional products, and life sciences chemicals. And lastly, Jubilant Biosys, which is the business that my colleague Ramchandraya and I both represent on this call today. And this business is our drug discovery and development services business. In this business, we operate right from a target selection all the way to a candidate nomination for a small molecule drug discovery program. And our capabilities also include both non-GMP and GMP drug substance manufacturing at a kilo lab scale. Talking further about Jubilant Biosys, we are an innovation-driven CRO. This company has been founded in 2020. We employ more than 750 people, and several of our PhDs come with 10 plus years of experience in the Western world. Having been in business for so long, we have worked with several of the top 10 global pharma. Sanofi is one of our active collaborators that is publicly known. And of course, we have a battery of customers who come from the mid-pharma and the virtual biotech ecosystem. We boast of a track record of having delivered on well over 75 integrated programs. Lastly, talking about our location, we have our Bangalore center, which is a fairly integrated center and operates across the functional areas of chemistry, computational, structural biology, in vitro, in vivo pharmacology, and ADME support, as well as toxicology. So here we run integrated drug discovery programs for our customers. The Noida Center is a chemistry-only center and has expertise ranging from milligram scale to multi-kilogram scale synthesis. At our Noida Center, we also have a GMP pilot plant or a kilo lab, wherein we provide GMP drug substances for our customers in multi-kilo scale. Lastly, we have also announced major expansion plans in 2019, and currently execution towards that is underway. So first of the block will be a Greater Noida Chemistry Center, wherein we'll have around 250 to 300 chemist capacity getting ready and commissioned for business in middle of 2021. And also we have acquired a sizable portion of land in Bangalore, where we'll be setting up a state-of-the-art integrated research center and a CDMO plant. With this, I'll hand over the baton to my colleague Ramchandraya Gosu, who is an alumni from Indian Institute of Science and has been leading our structural biology department.
Thank you, Saurabh. Myself, Ram. I'll take over to the topic of today. This slide captures in a nutshell what we do at the Department of Structural Biology. The major job is to support the integrated drug discovery programs in terms of reagents. That's enzymatically active proteins, co-crystal structures, and stables. For towards this, what we use is 3 different expression systems: E. coli, insect cell, and baculo, and mammalian systems. So we have in-house X-ray diffraction facility, and also we access the synchrotron at Australia as well as France. So we do use the biophysical characterization, that is thermal shift assay and SPR assays. A mass spec we access at nearby institute called Indian Institute of Science. We are expecting by next year we will have our own mass spec facility for the protein. So with that brief introduction about the department, I'll jump into the actual topic. So what are the platforms we have? Biophysical, thermal shift assay, and surface plasmon resonance based assay. So what are the readouts in this? If you look at thermal shift assay, what one does is we thermally denature the sample protein and find the melting temperature. The Cipro orange, typically there is a dye used to read the fluorescence. So once one finds the melting temperature. So in presence of the small molecular compound, the melting temperature shifts. That's what one does. Whereas in the case of surface plasmon resonance, the binding event is monitored over the time. So in this, you will get a real-time information. The resonance signal is plotted against the time. So if you compare these two techniques, what we know, readout is a qualitative in the case of TSA, whereas in the SPR, it is a qualitative as well as quantitative. And it is probe may interfere in the case of TSA and very subjective. The reliability is very subjective. Whereas in the SPR, 100% label free. So there is a very high impact for the medicom team to design compounds when we use the SPR data. Very minimal information we have in the case of TSA. So when you look at in the drug discovery lead cycle, it minimizes in all stages when we with the SPR data. Whereas in the case of TSA, the data doesn't give much clear information. It's difficult to differentiate actually whether heat or lead. Nevertheless, thermal shift assay has advantages when we handle the small molecule drug discovery programs. So I'm going to give a few examples how this impacts in small molecule drug discovery. So the first thing is it definitely distinguishes between the surface binder and the pocket binder. So what I'm showing here is for a case, for one case study where we have a surface binder. What I plotted here is temperature on x-axis, y-axis the first derivative of the fluorescence, that is shown in negative. So the different curves, what colored are I mentioned, what are those? So apoprotein is a dotted line. When you increase the concentration of the compound dose dependently, what we notice is there is a shift in the melting temperature towards the left in the case of surface binder. When we look at the pocket binder, in this case, two examples we have taken, one weak binder and the strong binder. Please note here, IC50 written is greater than 30 micromoles. It's not from primary assay, it's from the secondary assay information. So we know that it's a binder. So when we examine these two cases, that is a weak binder and the strong binder, what we find is the temperature difference is, melting temperature difference is positive. Whereas in the case of surface binder, the delta Tm is negative. That is the critical information one can get in thermal shift assay. I'll show another example where the cofactor, necessity of the cofactor for the compound to bind. What I mean to say, the inhibitor binding is enhanced by choosing the correct cofactor. So in this case, there are two cofactors, SAH and the SAM. So when you use the SAH, and I given the two curves here, one is the melt curve, on the right side you have a melt peak. So whenever we use the cofactor, what happens is the initial fluorescence is stabilized. That's what we observe. But when we do this first derivative of this and check the melt peak, what we notice is that the Tm shifts towards the right only in presence of SAM. So this clearly says what cofactor is required for the compound binding to happen. So this is one other piece of information one can get from thermal shift assay. So there could be, when we go to the fragments, it will be challenging to typically find the delta TM. That is a disadvantage. One more disadvantage is whenever is a protein is a multimeric enzyme, then if all the monomers doesn't uniformly denature, then interpreting the data will be difficult. So that's about the thermal shift assay. Now we'll switch to the surface plasma resonance staining platform. So I'll just briefly give about the principle and what is the technique it is used in the system what we have that I'm going to explain in subsequent slides. So the first, what we have the system at our Jubilant is called Pioneer FE, that's pictured here. And the principle here is, the simplest principle is, it depends upon the total internal reflection. The total internal reflection changes based on the binding event happens at the surface of the sensor chip. So whenever there is a resonance between the incident light, polarized incident light, and the plasma, the dip in reflected intensity. There are two events happen: a dip in reflected intensity and change in angle. The change in angle happens whenever diffractive index changes. Diffractive index, how it changes, there's a mass transfer happens at the flow channel. So these are the two events happen. So if you see in the right side, what I'm showing is the dip in intensity, and whenever the resonance happens, there is a dip in intensity of the reflected wave. While the binding events happens at the flow channel, automatically the angle also changes where you see that resonance. So these two informations. So what we capture in the SPR is response unit, SPR response units. So one response unit corresponds to typically 1 picogram per millimeter square. That is the mass change at the flow channel. So in the response unit, what we get the association, saturation, steady state, and dissociation. So when binding is complete, it reaches a steady state. Upon that, when we inject the buffer, it dissociates. So this is the response curve one gets in SPR. From this data, what we can derive is kinetic information, K on, K off, and KD. That means how fast it associates, how long it stays is the K off, this information. The ratio between the K off and K on is directly equivalent to the KD, the dissociation constant. So 1 over K off gives the resistance time. So this is the information. So what we get is the label-free technique. What we get is real-time information, K on and K off, and the equilibrium constant one can derive from that. So this is the data one can generate from the surface plasma. Now, what is the specialty? Before going to the specialty, let me touch upon how we immobilize the protein. So what are the chemistries involved in this? The most popular one is amine coupling method. The amine coupling is a irreversible method. It covalently the protein attaches to the gold surface through dextran molecules. So on the gold surface, we have a dextran molecule. We use the EDC and NHS2 chemicals to activate the gold surface. And subsequently, the ligand, what I mean ligand is a protein, which has the free amines, which forms a covalent interaction with the dextran molecule. So one disadvantage is if you have a protein and the active site license are there, it's not advisable to use the amine coupling method. One more preventive action we have to take is whenever you use this type of chemistry, we are subjected we are subjected to the pH shock. So we should make sure that protein is happy at the acidic pH. That one has to be take care. The another method is stepped out in biotin capture. In this, the dextran molecules, what we do is the streptavidin is a protein. It's a tetravalent for the biotin molecules. So streptavidin is covalently linked to the dextran surface, and the protein is biotinylated. So this binding is reversible and order of 10 power minus 15. Whereas in the case of amine coupling method is irreversible. This biocap method is reversible. So popularly both techniques are used for coupling the protein onto the surface. Now we look at the OneStep how it works. This is the specialty of the Pioneer FE system. What I showed in the right side is the biosensor. I just marked the circle. That's what the channel looks. 1, 2, 3 channel. This biosensor looks like. That means each 1 and 2, 3 refers to the sensors. What we discussed previously. So what we another picture I have shown is the impression on a filter paper. When you magnify 25 times, you can observe S-type diagram there. So before the when we inject a analyte, before reaching to the sensor, there is a capillary. The capillary is filled with the buffer, and the sample is injected subsequently. So this nicely forms a sigmoidal concentration gradient. So what I mean to say, at time t start, you have a 0 concentration. Over the time, concentration increases, and highest concentration is reached in single injection. So when we get the response curve, one response curve composite of multiple concentration of the analyte. That's the idea of the OneStep. So what we use is Taylor dispersion concept is used in this technology. So thereby diffusion coefficient can be measured. The diffusion coefficient directly related to the concentration. So if you use the 3% sucrose in the running buffer, that helps the interval control. So overall, the SPR assay to get in place, it takes what are the stages involved? The first is the preparation stage, having the protein ready. SPR, I mean, once make ready, we should make sure some biochemical assay in place. And the optimization involves one is optimizing the protein immobilization and finding the suitable buffer and validating with the reference compound and ready for the screen, and it's ready for screen the compounds. So the one important thing we have to remember is if you can reduce the non-specific binding, then we are successful in SPR assay. So once it's all ready for screen the fragments, 1,000 samples can be run in 72 hours without stopping the equipment. Unattended runtime is possible with this equipment. So the target which we prosecuted for this screening is it is involved in liver disease. It's a dehydrogenase enzyme. It is first in class. No structure is known. All the protocols we established producing for recombinantly now. Biochemical assay is established, and whatever required for the SPR assay, the protein is produced. Suitable protein is produced. Currently, structural work is in progress. So I just captured here in this scheme. What we find is this panel. What is the purity of the protein? Just to capture the purity of the protein. And the final protein, what we used is active, and signal to noise ratio, what we get in the biochemical assay tenfold. And prior to taking to the SPR screening, we need to have a positive control that has been established using the biochemical assay as well as partnership assay is one of the components. Now we'll spend some time how this screening strategy worked out for the fragments. So computational chemistry team collected 44 library. From that, 0.1 billion fragments are populated. From that, diverse fragments are pooled. That comes around 20,000. When you apply the filters, whether the fragment is natural product like, or does it have the sp3 centers, by filtering the and then made the clusters, atom pair fingerprints are used to further filter. So eventually we arrived at 2,000 fragments. Those are ready for the screen. So when we did the SPR screen, so that is called the point studies that I'll explain to you in the next slide. So now from the sensorgrams, we are able to get what are the binders, what are the non-binders. So whenever the ligand efficiency is better than 0.3, that we consider as hits. So we can segregate the structures. I mean, what I mean to say is based on the appearance of the moieties, we can decide as well as the molecular weight, whether it has to go to the full assay, all that we can do. So full kinetics one can run, and the computation assay also one can do. If you know substrate is known, whether the given fragment is binding substrate pocket or not, one can do in this SPR itself, SPR assay itself. So once we have the final prioritized confirmed hits, now this SPR up to this, what we know is as it binds or not, that information we know, and which are the good binders, that information we know. Now subsequently where it binds, that's where the subsequent work happens. The dotted line, what I show is the work in progress. So It is optional whether the fragments to take into the in vitro assay. Based on the size, one can decide. And of the two techniques, X-ray crystallography and NMR, what we chose is X-ray crystallography at present. Once we get the information about where it binds, that helps the multiple structures of the fragments where it binds. We know whether to merge those fragments or link it or grow further. It can be done from the computational work. And subsequently, it goes to the medicinal chemistry, and it goes typically it goes into cycle. Design, make, test cycle. That's how it goes. So what one required for this screening is one positive control definitely required. Negative control can be chosen as a buffer. Now I'll just explain how the bind studies work. So these are the sensograms one can get. So what I show in the blue color are the positive control sensograms. Green color are the hits. So there is one representation, actually one plate. When you use the data and duplicates, 192 fragments are there. How you analyze this data? So if you look at the positive control, at 60 seconds it starts to saturation is reached. So around that point, what are the responses we got the fragments? So those are populated here. X-axis cycle and the responses we got for each of the fragments. So now this data, what we have to do is we have to use the statistics called box statistics. In this, one has to find the Q1 and Q3, quartile 1, quartile 3, and we have to find the outliers. From the Q1 and Q3, if interquartile range is greater than 3, those are the hits. That is the idea behind it. So once we find the outliers, those are nothing but hits. So the hits are marked here in circles. So now those hits, what we did is we calculated the ligand efficiency and the KD on the y-axis. Whichever is greater than 0.3, we consider as a top priority. That we got 34 hits. And what I given the table here is the hits based on the moiety distributed. It can be classified into 8. And what are the numbers we got? I have listed here. So if you overall, if you see, we got a 2.5% hit rate we got when we started the 2000 fragments. When you look at the functional groups in the moiety, in the fragments, this can be further clustered only to the 6 moieties. So far what we know is as the fragment binds or not, that information. No? So definitely we need to have the crystallography where it binds. So being a novel enzyme, we started with the 24 constructs. We designed 24 constructs, and of that, we chose the 2 expression system. From this, we got only 3 versions, which are crystallization grade samples. Very first attempts, we got 7 angstrom data, and this took 3 weeks to grow. This is the crystal here. And iterative microseeding, this reduced the crystal harvesting time to 1 week. And additive screening, co-crystallization efforts with multiple compounds, rigorous in-house X-ray screening helped us to shortlist the few additives. And regular shipment to Australian and as well as Soleil synchrotron helped us to arrive at the 3.5 angstrom data right now. And right now, co-crystallization experiments are in progress with the hits. This is the crystal which helped us to get this data. So further work is in progress in terms of the crystallography. In a nutshell, what I can say is the biophysical platform, we can handle the real-time kinetics for both reversible and irreversible. We can perform the drug competition study. One can do the high-throughput screening, fragment screening, and we can identify the hits in a short period of time. With all that, one more information one can get when we do deal with the real-time kinetics. More than two compounds, 2 fragments or 2 compounds can have the same affinity. How you distinguish them using the kinetic data one can do. That's what plotted here. An x-axis association, y-axis dissociation. In the slant, in the slope lines, what we have is the same KD values. So, a compound, any 2 compounds having the same affinity, we can further deep dive and understand how one can characterize. So, thank you for your attention.